Well, good day, everybody, and welcome to today's Weber teleclass. My name's Martin Kiernan, and it's my great pleasure to host this teleclass. Our speakers are actually from this side of the pond over in the UK today. So, first, I'm going to introduce Helen Rickard to you. She's a doctoral student. She works in the Healthy Infrastructure Research Group at University College London under the professorship of Lina Siric. Helen's got her undergraduate degree in biological sciences from Lancaster, and then her master's in medical microbiology from the London School of Tropical Medicine. Before joining UCL, she worked for the UK Health Security Agency, conducting research into the role of different aspects of the built environment in infection transmission, and as part of the COVID research response. But her PhD that she's currently undertaking and nearly finished aims to study the bacteria found within hospital sinks and further understand these as a source of potential pathogens. But opening up is a good friend of mine, Professor Elaine Cloutman-Green. Now, Elaine is what I would call a really good example of a clinical academic because she comes from a non-medical background. She's a clinical scientist. She's well recognized as a foremost person in this field, to be honest, and as a real trailblazer. She works at Great Ormond Street Hospital, but also has a professorship in the same group at University College. She's been recognised nationally with the Order of the British Empire Medal. 2 years ago, the Healthcare Infection Society awarded her the Early Career Award for outstanding innovation contributions to prevention and control of infection. She's published many papers related to the environment because that's a specific interest of her. She chairs the Environment Network here in the UK. But also, interestingly, she's an accomplished playwright. She has a play that you can see online if you go looking on YouTube called Nosocomial. And she's dedicated her life to trying to communicate and get the message over. She also has a fantastic blog called Girly Micro. So do check that out as well. So she's a great speaker. Very much looking forward to hearing both of them speak as we listen to the session called To Aeruginosa or Not to Aeruginosa. So taking off Shakespeare, I think there. How significant are Pseudomonas in waterborne healthcare infections? Welcome, Elaine. Welcome, Helen.
Thank you so much, Martin. That's quite the intro to live up to, but we shall do our very best. So I'm going to kick off a little bit just to talk about really how this work came about. In 2009, I've been in infection control for a while, and we were looking at our first foray really into how design impacted microbes and the microbiome of the built environment. And we did some work in our intensive care unit, comparing it with a different intensive care unit. And the different intensive care unit was an adult medical and surgical unit. And we looked at how hand hygiene, because obviously I kind of get fired, I think, if I don't mention hand hygiene at least once in any infection control talk, was impacted by sink placement and sink visibility. And that work really kind of showed that if sinks are more visible, people unconsciously actually spend a longer amount of time washing their hands. And that was really interesting to me because they had like 3 sinks in a very like traditional Nightingale layout, whereas we had, because we had a new build, moved to 1 sink per bed space. And so at that point, all sinks should be pretty visible, right? And so we then repeated that work in our unit and found that even though we had 1 sink per bed space, people would step out of the sink in their bed space if it was a 3-bedded bay and move to the sink that was in the middle because it was more visible. More surprisingly, we also looked at the microbes on those sinks because we wanted to see how that frequency of hand hygiene impacted the organisms that we recovered. And we found that those sinks that were used less frequently actually had higher levels of organism on them, despite the fact you'd think that because you were hand washing more and depositing more organisms, that you would get more organisms on the sink. Because you're not having the amount of time in between episodes for the organisms to grow back up and to populate, you don't actually get as high a level of organism distribution on those sinks that are used frequently. So it has not just hand hygiene compliance benefits, but it also has some level of environmental control benefit. The thing that shocked me at the time was that 60% of the back of my sinks had fecal flora on them. So that meant that we were having a scenario where, because we had nowhere for people to put things down, people were putting things on the back of the sinks, then undertaking hand hygiene, picking up the contaminated item, and then moving to the next sink. So that was the start of our interest in sinks. And I think we'd done a lot of work looking at things like Klebsiella. And if we get a Klebsiella acquisition, sinks are always our first port of call. But we also spend a lot of time looking for Pseudomonas aeruginosa. But clinically, I work in pediatrics. I have a lot of big respiratory units. I know that Pseudomonas aeruginosa isn't the only kind of Pseudomonas that clinically I'm potentially interested in, clinically that my patients have. And so when we started this work, we really wanted to look at what happens when you have a completely new unit opening that shouldn't have any patient organisms in there. What happens to that colonization over time, and what kind of organisms do you get? So I'm going to hand over to Helen, who did all the work to really start to look into this and answer these questions to see whether actually the non-aeruginosa is an important thing in this patient population. Over to you, Helen.
Thanks, Elaine. So in the middle part of this talk, we're going to look at the role that sinks play in the transmission of healthcare-associated infections. We're going to touch upon the different Pseudomonas species and the reasons that they are such important causes of healthcare-associated infections. We're then going to look at the impact that patients may have on the bacteria found within hospital sinks, and touch on some of mine and Elaine's work. And then I'm going to hand back to Elaine as our clinical expert to talk about the importance of having an interdisciplinary approach in infection prevention. So what are healthcare-associated infections? And there are variations in this definition, but in general, these are infections which have developed due to being in contact with healthcare or a healthcare setting. And so this could be something like developing a surgical site infection following an operation, or developing Clostridium difficile infection from spending time in an aged care facility. And healthcare-associated infection rates vary across the world. In the US, the CDC has estimated that every day, one patient in every 31 hospitalised will contract an HCAI. And in Europe, it's thought to be about 7% of healthcare patients will develop one of these infections. And the risk of developing an HCAI is higher in middle and low income countries. And interestingly, modelling studies have suggested that the global rate of HCAI is increasing by about 0.6% each year at the moment. And these infections have huge human and financial costs. And so these financial costs are due to the extended time that patients will spend in hospital, prolonged treatment durations, and the use of last resort antimicrobials, as well as infection control measures such as isolation and enhanced screening and cleaning. And there will also be indirect costs to the economy caused by these infections, which typically come about from loss of productivity in both patients and staff unable to work. Oh, wrong arrow. So HCAI can affect any organ or tissue type, and there is huge variation in the prevalence of different types of healthcare-associated infections depending on the geographical location and patient characteristics. So anyone that's been exposed to a healthcare environment can catch these infections, whether you're a patient, a visitor, or a staff member, but they are more prevalent in people undergoing invasive treatments or with other underlying vulnerabilities. And healthcare-associated infections can be caused by endogenous organisms or exogenous organisms. And endogenous ones are the organisms that naturally live in or on someone, for example, the bacteria in your gut or on your skin. And these can cause disease if they move to a different area, for example, a wound or the bloodstream. whereas exogenous organisms are those that come from external reservoirs, including other patients or the healthcare environment. The healthcare environment is a significant reservoir of HCI causing organisms, and the patient environment includes air, water, and surfaces around someone, and contamination of all three has been linked to the transmission of microorganisms resulting in healthcare-associated infections. And a recent review has suggested that the aqueous hospital environment, so that includes potable water, taps, showers, sink surfaces, along with drainage systems such as traps and toilets, is the biggest source of multi-drug resistant gram-negative bacteria in hospitals, and is likely a key origin of outbreaks. And as Elaine was saying, hand washing is well understood to be key, and some would say the most important intervention in the prevention of healthcare-associated infections, as it reduces the transmission of organisms from surfaces and between people. However, organisms are bacteria are able to reside in sinks that are used for hand washing, and these have now been implicated in several outbreaks. And sinks are a unique component in the hospital environment, so you need them to wash your hands, but they are at the water surface interface and are a connection between patient, staff, and plumbing systems. They're also a unique physical sort of attribute of the environment, as they're really hard to clean. So cleaning products and disinfectants will run through, and so inhabiting microorganisms won't be exposed to the necessary contact times. And sink components such as drains will be physically inaccessible for cleaning. And this allows the formation of biofilms, which promotes organism survival. And sinks also have a mechanism of dispersing these organisms. So the forces generated by running water from taps are able to produce droplets, which can contain bacteria. And possibly due to a cognitive association with cleanliness, so we wash our hands and that makes us clean, so we think that sinks should be clean. Sinks have really been overlooked in terms of infection prevention and control recently, and it's only recently that their role in outbreaks and infection has been realised. And so in the last 15 or so years, there's been a number of infections and outbreaks caused by bacteria from hospital sinks recorded in the literature, and I've just put a few up here. Oop, too far. And the number of infections caused by sink-associated organisms is likely to be much greater than we currently realise due to the transmission of drug-sensitive organisms and non-outbreak infections. And there are some key themes seen in these publications. And so first of all, it's the organisms that are causing infections associated with sinks. So the organisms that live in sinks and the associated plumbing and that are able to cause infections include both the normal microbial inhabitants of drinking water and those that have been deposited in or near sinks by human activity. So opportunistic premise plumbing pathogens, or OPPPs, are pathogens and normal inhabitants of drinking water. So they're not contaminants, but they are adapted to grow and persist in drinking water systems. And this group includes organisms such as Legionella, Mycobacterium, Pseudomonas, Acinetobacter, and Aeruginosa species. And microbes can also be deposited onto sink surfaces by human presence and activity, such as hand washing, toothbrushing, waste disposal, and certain medical procedures. And in the literature, these contaminants largely consist of gram-negative bacilli associated with the human gut, such as Klebsiella and Enterobacter species. And organisms within both of these groups share features such as the ability to survive in low nutrient and temperature areas, to form biofilms, and to resist low levels of disinfectants. Another theme tends to be the patient groups affected, and so as with healthcare-associated infections in general, the patient populations most affected by sink microorganisms are those with underlying vulnerabilities such as neonates and those undergoing invasive treatments such as central lines, or therapies like immunosuppression. Another key theme within these outbreaks is that they often need interdisciplinary and multimodal approaches to end them. So some outbreaks will be controlled by sink cleaning and disinfection, with chlorine-based cleaners typically being the most effective. However, in some outbreaks, the causative organisms have been found to be highly tolerant to the disinfectants or biocides used, and the full replacement or removal of sinks and associated plumbing has been necessary. And this is often seen where the organisms are residing further within the plumbing system, typically within biofilms. Installing barriers around sinks and moving medicine and device preparation areas away from sinks has also reduced the contamination of both the sinks and the medicines and devices, and been a key way to end outbreaks and reduce infections. And educating staff, patients and parents has also played a critical role in ending some outbreaks. For example, teaching or reminding staff not to dispose of waste in sinks and asking patients and parents not to leave personal items such as toothbrushes near them. Okay, and in the first part of this talk, we have gone through how important hand hygiene is as a preventative measure. However, the sinks that we need to wash our hands in do allow biofilms to form and have a mechanism to disperse organisms. And the organisms that are linked with sink-based HCI can come from both water and human activity. So what or who are Pseudomonas? Pseudomonas are well-known causes of healthcare-associated infections. They're gram-negative bacteria which are able to colonize a huge range of environments within both the natural and built worlds. So a quick look in the literature, if you put Pseudomonas into PubMed, you'll find publications on fish, and then you'll find things on hospital sinks. And this is highlighted by the Pseudomonas' ability to persist in damp environments, which allows them to colonize areas such as sinks, drains, and pipes within hospitals. The genus Pseudomonas includes over 140 species, and more than 30 of these have been shown to be able to infect humans. And the Pseudomonas species that are most commonly associated with human infection are Pseudomonas aeruginosa by quite a long way, and then other species including Pseudomonas petida and Stutzeri. And these species are such successful pathogens or opportunistic pathogens due to their ability to survive in a range of environments, to resist antimicrobials, and to produce a range of virulence factors. So Pseudomonas aeruginosa is the most well-known and clinically relevant Pseudomonas species. And I think this is a UK statistic, but it's the fourth most frequently isolated nosocomial pathogen, and is thought to cause up to 10% of all healthcare-associated infections. And Pseudomonas aeruginosa is able to colonize or infect nearly all, pretty much all tissue types, and so infections caused by Pseudomonas aeruginosa include urinary tract infections, wound infections, pneumonia, and bloodstream infections. And Pseudomonas aeruginosa can be extremely drug resistant, so strains have developed resistance to multiple antimicrobials, with some isolates now expressing resistance to nearly all available antibiotics. And in 2017, in the US, multi-drug resistant Pseudomonas aeruginosa caused over 30,000 infections and resulted in almost 3,000 deaths, alongside significant financial costs. And carbapenem-resistant Pseudomonas aeruginosa has been classified as a high priority pathogen by the World Health Organization due to its ability to develop and spread antimicrobial resistance alongside the threat it poses in healthcare settings. Sorry. Another factor that supports Pseudomonas aeruginosa's sort of ability or notoriety as a cause of healthcare-associated infections is its ability to persist in the environment. Isolates are often strong biofilm producers and are able to survive in damp areas for extended periods, but have also been shown to survive for up to 48 hours on dry surfaces. And this persistence means that Pseudomonas aeruginosa is well adapted to survive within plumbing systems, and is one of the most common common causes of HCI transmitted through healthcare water systems, with a Canadian study even reporting that 7% of healthcare-associated infections caused by Pseudomonas aeruginosa were actually associated with sink-to-patient transmission. And so now onto our non-aeruginosa Pseudomonas. So despite being less commonly implicated in disease than Pseudomonas aeruginosa, other Pseudomonas species are known to be able to cause a range of infections, particularly in children and other vulnerable patient groups. And a large proportion of infections caused by these non-aeruginosa strains in the literature are healthcare related, including those caused by contaminated drugs and equipment. And the numbers of infections caused by non-aeruginosa Pseudomonas aren't particularly well documented, but they are increasingly documented, with Pseudomonas putida recently having been called an emerging pathogen of healthcare-acquired or associated infections. And so non-aeruginosa Pseudomonas increase healthcare HCI risk and severity as they're found to be highly drug resistant and able to transfer these resistant determinants to more traditional pathogens. So they're able to contribute to the overall burden of healthcare associated infections by driving antimicrobial resistance, even if the numbers of infections aren't particularly high in themselves. And this is also highlights another role of sinks in HCAI. And so sinks are areas which support the development of multi-species biofilms, and we might see the transfer of antimicrobial resistance determinants between our more environmental organisms and those that are more able or likely to infect humans within sinks. In general, much less is known about non-aeruginosa Pseudomonas species in terms of location, biology, and virulence potential, despite the recent increases in human infections by these organisms. And virulence factors are sort of components or factors that allow an organism to infect a host and are often responsible for disease symptoms. And these include toxins and degrading enzymes, as well as the secretion systems which can deliver these factors to host cells, motility and adhesion determinants, and the ability to produce biofilms. And if you look in the literature for Pseudomonas aeruginosa and virulence, I think it's 7 or 8 papers that have either used the word arsenal or war chest to describe the numbers of virulence factors that this organism can produce. And there are numerous studies that investigate the virulence potential of clinical isolates of Pseudomonas aeruginosa, but there is comparatively little research on environmental Pseudomonas aeruginosa and pretty much none on non-aeruginosa species. And so this is just a quick snapshot of some of the virulence factors that Pseudomonas species, in particular aeruginosa, are known to produce. So Pseudomonas aeruginosa isolates nearly always produce exotoxin A, which contributes to severe infections and disease symptoms, and this is through interrupting host protein synthesis pathways. This has only ever been seen in Pseudomonas aeruginosa, as far as I'm aware, not the non-aeruginosas. But a huge range of Pseudomonas species will be pigmented, and these pigments can act as virulence factors by acting as siderophores to gather iron from the environment or the host, and they can also provide protection from light or the immune system. And then we have enzymes such as proteases and hemolysins, which can aid bacterial invasion by breaking down host components such as connective tissue or blood cells, and so can also progress disease symptoms. Pseudomonas biofilms are important both within the built environment and within the patient, and they help organisms persist by providing protection from cleaning antimicrobials and the immune system. Pseudomonas isolates are often motile, and so they rely on structures such as flagella and pili to move through and on aqueous and solid surfaces. And as well as being critical to motility in both the environment and living hosts, these structures have also been implicated in pathogenesis. So in mice studies, having an identical strain, two identical strains, one which will have flagella and one which won't, the flagella-producing strain will have a much higher fatality rate in the mice. And the pili that have been involved in moving have also been shown to be able to transfer genetic material between species. And virulence factors can be involved in both environmental survival and disease progression. And so from this little section, we've seen that Pseudomonas species are ubiquitous in the built environment, particularly in aqueous areas. And Pseudomonas aeruginosa is a leading cause of healthcare-associated infections globally, and this is likely due to its ability to survive in the environment, to resist antimicrobials, and to produce a range of virulence factors. And much less is known about our non-aeruginosa Pseudomonas species, despite their increased role in healthcare-associated infections. And it's now widely accepted that the environment is an important cause of healthcare-associated infections. And there is evidence of organisms being transmitted from air, water, and surfaces to patients, resulting in HCAI. And in some cases, the organisms causing these infections may naturally reside in the environment, such as Legionella species. And in other cases, the organisms likely entered the environment from other patients or from human activity. And really interestingly, several studies have highlighted that there is a significantly increased chance of a patient becoming colonized with a specific microorganism if the previous occupant of the room was colonized with it. And this is being demonstrated really nicely with some of the multi-drug resistant pathogens. And so, sort of pulling all of this together, I'm going to now briefly touch on some of our group's research investigating the impact that humans might have on the types of bacteria that live within hospital sinks and the virulence factors produced by the Pseudomonas isolates that are found there. And this was one of the first and one of my favourite parts of my PhD, so I hope you find it interesting as well. So swab samples were collected from sinks across 2 new wards at Great Ormond Street Hospital. And as we are interested in the impact that patients have, swabs were collected 1 week before and 8 weeks after these wards opened to patients. And then after opening, the wards served quite different patient populations as well. So Ward A was used for post-surgical care, and Ward B was used for long-term respiratory support. And then the resulting organisms from the swabs were identified using chromogenic agar and MALDI-TOF technology. And following identification, clinically relevant organisms, and so that was anything that had UCAST breakpoints, and all of the Pseudomonas recovered were tested for antimicrobial susceptibility. And all of the recovered Pseudomonas isolates were tested for the production of a panel of virulence factors using phenotypic assays. So we recovered a total of 106 isolates, and 75 of these came from the later time point. And so we saw changes after the wards opened to patients, and then sort of between time 0 and time 9. And then at time 9, we also saw differences between the 2 wards. So before the wards opened to patients, fairly similar organisms were recovered from both wards, with the environmental bacteria Cuprivardis, Stenotrophomonas, and Pseudomonas species being the most frequently isolated organisms. And then we saw increased diversity in the organisms recovered after the wards, and differences between the 2 wards. So in the isolates recovered from Ward A, we saw a shift from Cuprivardis species being the most prevalent to gut flora, including Enterobacter and Klebsiella species being the most prevalent. Ward B also had an increase in this gut-associated microorganisms, and also in the number and type of Pseudomonas species recovered. In particular, we saw a large increase in the number of Pseudomonas aeruginosa isolates after Ward B opened. And you can't really see that in this graph as it's just to genus level. So you have to take my word for it here or read the publication. And so we had a total of 24 Pseudomonas isolates, which were recovered, including 5 Pseudomonas aeruginosa strains. And of the 24 Pseudomonas, 71% and all but one of the Pseudomonas aeruginosa were recovered from the later time point. And this supports the idea that the changes in bacterial species were potentially due to patient influence. So Ward A may have had such a high proportion of gut commensals, as most patients in there would be in nappies, and so if carers or staff were changing nappies and then contaminating and then washing their hands, that could explain the acquisition of this fecal flora. And in Ward B, like hand washing after toileting, but then as a respiratory ward, there was a cystic fibrosis patient population here who would be likely to be colonized with Pseudomonas species, and that could explain the increase we saw in Ward B. And then in terms of antimicrobial susceptibility, the majority of the isolates we recovered were sensitive, and of the small population that did show resistance to any of the tested antimicrobials, 91% of these came from the later time point, which again potentially suggests there is patient influence here. So are the more resistant strains coming from patients? Is that this due to sort of extended antimicrobial therapy, particularly in like the long-term cystic fibrosis patient population? Or it could be coming from pressures that are seen within the sink, so disposal of clinical waste or the use of biocides and disinfectants. We then looked at the ability of the isolated strains to produce virulence factors. And so of the 24 strains isolated, 25% produced pigment, 58 were hemolytic, so were able to break down blood cells. 23% produced alkaline proteases and 8% produced gelatinases, and these are examples of the enzymes that will break down different types of human tissue. 83% twitched, which means they were able to move on solid surfaces, and 88% could swim, so could move through aqueous surfaces, and 71% produced biofilms. And so the images on the top show the virulence factors, which are more associated with host invasion and disease. And the images on the bottom show the virulence factors, which will be associated with both pathogenesis and environmental survival. And our results sort of largely fit with the literature, although we did see slightly lower levels of the disease-associated virulence factors. And this is likely due to the environmental origin of our strains. And there's just not that much in the literature looking at non-Aeruginosa strains, so it's quite hard to compare these. And we found that isolates recovered from the later time point were more likely to produce virulence factors. And in particular, they were more likely to produce the virulence factors shown at the top, so associated with human infection and disease, whereas the rates of swimming, twitching, and biofilm production were largely consistent over time. And another really key finding is that we showed that non-Aeruginosa strains were able to produce virulence factors that are typically associated with Pseudomonas aeruginosa, and so highlighting the role of these organisms and other non-traditional pathogens to cause healthcare-associated infections. And so both aspects of this study, so when we looked at the types, the susceptibility profiles and the virulence of the organisms found, add weight to the concept that patients likely are influencing the bacteria in their environments, including sinks. And I'm going to hand back over to Elaine now to talk about the clinical impacts of this and the importance of interdisciplinary work in infection prevention control. Thank you.
Thanks, Helen. So I suppose one of the questions that Helen and I talked about a lot when we were putting together the paper is the so what aspect of all of this. Does it matter if these organisms aren't particularly resistant? Does it matter that we are not necessarily used to looking for them within clinical environments. So what does it mean now that we're finding them? I think for me, a lot of this is about how we inform our risk assessment strategies. Within the UK, I think there is an increasing recognition of the impact of non-traditional pathogens in impacting healthcare outcomes. So many of you probably may or may not know about things like some of the incidents that have happened in hospitals where things like Cupovardis have been demonstrated to have significant patient impacts. And those are not the kinds of organisms that would normally end up triggering an outbreak alert, a big infection control response. Often we wouldn't necessarily even be handed over those kinds of results. I think it's worth saying for context, just because I have been to other places to work in other countries where infection control works quite differently. Within my practice in the UK, all positive results will come through the infection control team. So we will see everything. And we hold clinical responsibility for therefore choosing interventions and actions based on those results. I know that it does work differently in other places. Partly that's because of the fact that we also have a system of governance that requires the Director of Infection Prevention and Control to have delegated ownership of things like water and ventilation issues from the chief exec. So it's embedded in our guidance, it's embedded in our responsibility, and we have other guidelines linked to things like the BSI standards that state that we should be doing regular water reviews, that we should be looking at organisms much more widely than the traditional Pseudomonas and Legionella. And that's built on the ever increasing evidence set that shows that outbreaks do occur with these other organisms. So it's important to have this review process. So every month I will sit with my team and we will do things like trend analysis for all of our non-Aeruginosa pseudomonads, for things like our Stenotrophomonases, for our Elizabeth Kingias, our Burkholdeриas, to be able to see whether we have something on the horizon outside of those more traditional pathogens. Whereas I said at the start, if I get a hospital-acquired Klebsiella, I will go and do things like screen my sinks in order to identify it. And in some ways, we've learnt the hard way about non-Pseudomonas aeruginosus, in that I have had clinical cases that took some time to unpick and flag, that were then identified linked to sinks and sink positioning. And a very classic one was in a very old ward where the sink is actually much closer to the head of the patient than you would design now. And I'd even flag to you that most of our guidance says a meter away from a head is enough, whereas the ever-increasing literature shows that we really need to be looking at a 2-meter splash zone that should be clear of all clinical equipment. And it takes our guidance some time to catch up with that. So being invited and being asked to speak at webinars like this, I think, is so important, because as infection preventionists, we need to be having those conversations before the guidance comes out, because our buildings will be taking years to be designed and built, and then they will stand for decades. And so if we are not putting those things forward in terms of conversation, then we can be left with a building and a risky environment for some time. One of the other things that I think is so key in terms of what Helen has shown is that we often think of the built environment leading to patient acquisition, and the fact that things will come from the water and may splash onto a patient, and it's a one-directional transmission. I think this data shows very clearly that our patients influence their environment as much, or if not more, than the environment influences your patient. It's just your environment is then a long-term presence as those patients change. I think that's particularly key in terms of thinking about interventions and practices in populations like mine, where I have children who will not use sinks as you might expect. A 4-year-old is not going to stand in the right position undertake hand hygiene the same way that a trained member of staff would, and yet we often interpret the risks in the same way. I also will have patients that will stay for, some of them 5 years in the same space. So that establishment of their flora within their environment is going to be considerable. And as Helen has stated, removal is not easy. Even thinking about, and we spend a lot of time in terms of our interdisciplinary approach to this, talking and really spending time with our facilities team to think about how to clean a sink. I even ended up doing one for the Royal Society of Public Health, if any of you are particularly keen on thinking about sink cleaning, because it is multi-stage and complex, and many people don't necessarily pay sufficient attention to it because they believe that sinks are associated with cleanliness, and therefore many of your staff will perceive them as clean. In reality, if you want to be able to drop your organism loads, you need to think about things like drying your sinks before you put your agent on there, because your agent is all based on concentration and contact time. And without that combination, you're not going to get the safe environment and the clean that you think you're going to get. And if you're in a scenario where that organism and that patient flora is going to be heavily established because of length of time, then you may have to think differently about your cleaning in order to get rid of it. Traditional non-touch techniques like hydrogen peroxide vapor split into oxygen and water. If you have a dry sink and you think that that hydrogen peroxide vapor is therefore going to help reduce your loads on your sink, it probably isn't, unless you've dried it and it's in a position where you can have that physical reaction occurring on your surface. So even those things that we do to add extra confidence and comfort in terms of our decontamination techniques require additional thought when you're talking about an environment and a piece of technique and kit that is quite different. The other thing, and that I think us all getting into rooms together to talk about these things is so important, is because it isn't just sinks that contain water. So it's not just sinks that are going to contain this non-Aeruginosa risk for your patient population. In my world, people will go out and they will buy bubble tubes with water in them and put them in bed spaces to distract patients. And those bubble tubes are not sealed, and they will bubble air through water, producing aerosols and droplets that will then interplay in a similar way, but without somebody actively cleaning them, without somebody actively putting any form of chemical or agent into that water tube. And with an extra level of risk in terms of kids will get up quite close and inhale and really interact with them in a way that they're probably not going to do on your sink. And that's just one example of a water utilizing piece of equipment in our environment. And if you don't have that engagement with people like your play teams, with your nursing staff, with all of your healthcare assistants who may be taking these things out of cupboards to give them for interaction, then you may not see them when you're doing your risk assessments and your walk-arounds, in order to actually think about, A, whether they should be there in the first place. I love switching them out for solid light things that are easier to clean. But B, if you need to have them there, what can you do differently with them? I'll give you the example of, we had a parent who was incredibly keen on bathing her child every day as part of their bonding exercise, but they brought a water bath from a common internet supplier, and it had an elephant trunk, and so they would use the elephant trunk to spray their child because their child was incredibly amused by it. The problem is a lot of these things come preloaded with organisms, and so their child ended up having 2 rounds of bacteremias. Bonding and that social interaction is so important, though, when you have a really unwell child. So then it's about thinking about, is there something else that we can provide that would do the same job, that is safer, that is more cleanable? Is there a cleaning intervention that we can do? Or do we just accept the fact that maybe we have to throw this away much more regularly, maybe get rid of it once a week and absorb the cost of that in order for the protection of the patient, because a bacteremia is going to be much more impactful on their outcome and much more expensive to deal with than buying a £20 baby bath every week. And so we have to be inventive and pragmatic about our interactions once we understand how people are using sink environments and water in order to get the best possible outcome for everybody. The final thing I kind of want to talk about before we finish, really, in terms of how we need to think about sinks moving forward, is I really want to talk about the importance of an MDT in a slightly different way in terms of risk assessment and the role of things like risk assessment walkarounds that should include your estates team, your water guys who are going to actually be doing work with the sinks, but also people who are going to be leading on the cleaning, as well as the clinical teams and infection control, and ideally a patient representative, so that we have patient voice into all of this, to really think about risks and impacts, because I think all of us bring specialism and expertise, but the real magic happens when we come together and listen to each other and hear other people's input into why certain things matter, to then make us think about this entire scenario in a different way. Because water-related risk is not going anywhere. I know that people talk about things like water-free, water-reduced, and I think what we should be talking about is water-appropriate. If you rip out all of your sinks and you have a C. diff issue or a norovirus issue, or if in my case you have loads and loads of patients in nappies where you're going to get contaminated hands, that is probably going to cause you a different type of risk. But there will be environments where sink reduction is appropriate. And so it's all about appropriate use and not just sticking a sink in every bed space, which that original data that we did in terms of looking at sink use has probably shown is not the best plan, because low use sinks have higher microorganisms and low visibility sinks have less good impact on hand hygiene. Having vastly reduced sinks also impacts practice. So what we need to do is do an active reflection and put in that energy and engagement in that MDT fashion to make sure that we're having appropriate use, and that we think about positioning, and that we don't get bullied into accepting a meter when we know that the evidence shows 2 meters, that we're actively going out there and looking at what the data is to support our decision making, so that we end up with a water use that actually improves patient safety, that doesn't end up being a risk. And so there are roles for all kinds of things in terms of preventing and controlling outbreaks from sinks. As part of that risk assessment, doing things like microbial surveillance, and that can be testing your water to see if you've got bat contamination to your sink. It can be things like, as I said, if I get an acquisition of something like a Klebsiella, I will go out and actively screen my sink. Now, if I get a trending up in things like my non-Aeruginosas, I will then dive down into it and start looking about what people are doing in that environment much more. So it flags to me. I spend a lot of time thinking about what my disinfection processes are and how I do them. I have ended up spending fairly significant amounts of money in some cases in order to physically change both my sinks and my sink positioning in order to make them better. I will actively do things like remove fin sinks when I find them. And that's not because fin sinks are negative in all settings, but they work under very specific circumstances. So they have to be positioned appropriately, and you have to have people who use them in the right way. That's not going to happen if actually a lot of my sink use is linked to visitors who do not know how to stand appropriately, or if I've got children at various heights who are using them. And in those circumstances, we found that actually they increase risk, they don't decrease them. As I said, really thinking about that sink placement and numbers piece, and not ignoring sink use and maintenance. So we do things like we do sink audits to check sealant integrity, to check whether we've got any black mold, to check that the IPS panels are appropriate, because that's embedded not only in terms of an external audit, but we ask our sink cleaners to do that every day as part of their practice, to flag when people are storing stuff on the sinks, to flag when it's not looking its best, where the sealant is peeling away, because those are the things that can go unnoticed that then lead you to have an issue further on. And so education and training about the fact that sinks are important, that sinks are used for cleaning, but they are not clean, is a really important conversation and dialogue that we really need to embed in everything that we do. And so if we recognize the fact that infection control does not have all the answers, but we're prepared to step up and have a position in terms of leadership, in terms of governance and assurance, then we can have much better conversations about all of these pieces so that we complete the jigsaw puzzle faster when we need to. And this work that Helen has done has required huge amounts of her time and a lot of dedication. But I also want to thank the guys at Great Ormond Street who did the original sampling, the infection control team who do all of the data collection, to people like Elaine and the laboratory there who have provided so much training and engagement. And I just want to hand back to Helen in case she has anybody else that she would like to say thank you to.
Yeah, definitely. Thank you to the staff members who have let me come in and see the different sinks and the way that they work. And thank you to the patients and carers who've been there as well. And thank you to Paul and Martin as well.
Okay, I think this gives me a chance to hop in, doesn't it, on questions? So thank you very much. I really enjoyed that. It's absolutely fascinating, and it's a really fascinating piece of work and well worthy of doctoral level study. Couple of questions. Elaine, you say you go sampling your sinks. Should infection prevention teams be thinking about doing sink sampling? And secondly, would a clinical micro lab in the average hospital be able to do that, or is this something that ought to be outsourced? Because they're probably looking for organisms they haven't regularly seen.
Oh, that's a really good question. So I think that, and I'm talking about it because I've been talking about it a lot actually as part of the new guidance stuff that we're doing. So I wouldn't necessarily advocate for things like routine drain sampling, because to be honest, I expect to find organisms down my drains.
Yes,
because that's the
sort
reason
of the
people
point
are
of them,
washing
isn't
their
it?
hands,
Yeah.
right?
Hopefully.
So that's kind of so I find those really difficult to interpret. Things like and I expect that in the sink bowl, but I will also do things like back of the sink sampling, where if I find it there, it's either because of splash or cleaning failure,
Yeah,
and
it's
that
more
helps
relevant, isn't it?
me
Yeah.
interpret. So I sample sinks, but I sample them with caution in a way that I can interpret them. I think that luckily within the UK, most of our clinical laboratories could, and what we're currently working up, as you know, and I'm hoping that we continue to work on, is how we develop methods to support those laboratories actually interpreting them. It's one of the reasons that I feel like it's really important to use pragmatic sampling, so sampling with swabs and things that you have on site that the lab know how to process because they're used to processing them from patients.
Yeah.
And therefore, often the organisms I'm looking for are things like, I don't know, a Cupovardis, a Steno, that I'm also doing trend analysis for. So they should have the identification ability to do it. That said, I am aware that Gosh is a pretty special place, and that I am very fortunate in a lot of the techniques that we have. And so one of the things that I think we're all trying to work up is what is the best way to do this? Because I don't think there are a lot of environmental labs, to be honest, if people are used to sending stuff away, that necessarily have the same identification capabilities as a clinical lab, because they're used to mostly doing the big, large numbers of sampling for things like Pseudomonas and Legionella, and they're only just getting things like molded off. And so I think there's a drive to move the entire discipline forward together in order to do it better.
Okay, thank you. I was interested in the fact that when you did your initial sampling of Ward A and Ward B, you still had quite a lot of organisms there. I was wondering how long before opening, as you know, they were connected up to the hospital drainage system so that they could actually become preloaded with what looks like a selection of water type organisms rather than the gut organisms that come in later. How long was that? Any
It
idea?
was about a month. So
Okay.
so it wasn't a massive amount of time, but obviously you have to then commission the system and then take the water samples. Now, interestingly, although there was Pseudomonas found on the sinks in terms of Aeruginosa, the ward was Pseudomonas aeruginosa free in terms of its water at commissioning. So it's really interesting to me, like whether at the point that that building is handed back, people are then loading up cleaning buckets and things from places that you might not necessarily have tested, or is it that actually that pipework becomes fairly rapidly seeded with things like non-aeruginosas with some level of aeruginosa, because of the way that people are moving around and doing things, because the clinical staff will have been moving in equipment and stuff at that point, even though there were no patients. And so I think it's really interesting to me how quickly something goes in, or is it actually at manufacture that because they will test some of the sinks and things, even though we're not finding it in the water, is it that the sanitary ware almost comes preloaded from the factory?
Yeah, you do wonder that, don't you? I was fascinated to see an organism I'd never heard of before, Cupria viridis, there in big numbers before opening. And I was trying to rack my brains at my very schoolboy Latin, thinking, is this because it's a copper no, it can live in copper piping, so you're not actually flushing it much while you're pre-opening, and then once you started using it and everything else flushes through, does that disappear? So, was that preloaded in the factory with the copper? I did wonder that.
Yeah, and I suspect that the water doesn't have the same flushing rates because this entire building opened at the same time. I wonder if things like Cupovardis, which obviously you're not testing for in terms of your initial opening water testing, is there at high levels until you actually get your patients in, and then you're using your water much more and flushing those systems through.
Yeah. Now, my last question is about, as you mentioned, is some organisms you don't test for. And I'm just wondering about what's the risk of horizontal gene transfer from these environmental classed as non-pathogenic Pseudomonas and others, given that most clinical micro labs may well just dismiss those as a contaminant? Is there any evidence that they will? Because I know Acinetobacter radioresistance carries carbapenemase resistance genes, and you wouldn't report that. I've never seen a clinical specimen with that, but it could easily hand it over to a different, more significant Acinetobacter. Is that similar with Pseudomonas?
Helen, do you want to have a go, and then I can talk about the clinical incident we had?
Yes, I'll jump in here and say yes, definitely from what I've seen in the literature. So Pseudomonas petita has been found to transfer antimicrobial resistance determinants. And the conditions that are in sinks are also likely to sort of increase this. So I've seen a paper that's found that under like low levels of biocides, gene transfer is increased. So sort of like the combination of the two, having these highly drug resistant but not very notable organisms within sinks is definitely a gene transfer risk.
And I think we've definitely seen it actually happen in live clinical practice, Martin.
Well, if anyone has, you have, because you've been looking.
So I've seen it happen with the Pseudomonas putida that actually went on and colonized 2 patients. I had a big outbreak, not from a Pseudomonas, but from an Enterobacter that did gene exchange with a Klebsiella that then caused an outbreak that lasted over a year, to be honest, and then caused huge amounts of both patient disruption and financial implications in terms of ripping out sinks and things. And that's why I think the fundamentals are so key, like getting your sink cleaning right, getting your placement right, getting your testing and surveillance algorithms right, because you're never going to be looking for everything all the time. And so you have to get your first principles right to protect you, even when you're not necessarily searching for it, because there'll always be something new and surprising coming down the line.
Yes, always. Okay, well, thank you very much. That was absolutely fascinating. I've really enjoyed every minute of that, and I look forward to see further work from the group. If I could highlight the next teleclass education topics coming up, and next week on the 26th, we have a WHO teleclass looking at the global situation of infection prevention and control and the case for action investment in improving it. And that's Professor Benedetta Allegranzi from Switzerland and Dr. Michele Cecchini from France. And then moving on to March, there's some really good ones here with Michael Borges from Malta talking about preventing MRSA bacteremia. I can guarantee that will be an excellent teleclass. The next one after that will be the next pandemic. Are we prepared, with Professor Michael Klumpus? And I suspect the answer to that one will be a 2-lettered word rather than a 3-lettered one. As ever, we thank our teleclass education patron sponsors, Diversi, Virox Technologies and Gamma Healthcare, without whom teleclass education could not take place. And thank you so much again, Helen and Elaine. We really appreciate you giving us your time and sharing your expertise with us.